Thank you for joining us for this webinar on Design High Specificity CRISPR Cas9 gRNAs, Principles and Tools. My name is Heidi Huang and I'm the Service Marketing Manager for Molecular Biology Services at GenScript. Prior to joining GenScript, I completed my PhD in molecular biology at Vanderbilt University, working on DNA replication and DNA repair using simian virus 40 as a model system. As a basic research scientist, I'm always very excited to see discoveries from basic research get transformed into innovative applications in biotechnology and medicine. CRISPR is such a wonderful example. CRISPR-Cas9 systems are first discovered in bacteria as an adaptive immune defense mechanism that utilizes short RNA to guide degradation of viral DNA. Recent development of CRISPR-Cas9 has empowered it as the most wonderful genome editing tool for diverse organisms. In today's webinar, I will first give you an introduction of CRISPR-Cas9. I will start with its function as part of the bacterial adaptive immune system, and then review how scientists engineer the Cas9 system and exploit the host DNA repair pathways to achieve targeted genome editing. I will use mammalian system as the example throughout this webinar. In the second part, I will walk you through steps involved in gRNA design and provide detailed guidelines at each step to get high specificity gRNAs that avoid off-target effects. I will also introduce CRISPR services and resources offered at GenScript to help you achieve your research goals. If you have any questions during this presentation, you can submit them by typing them in the question fields that should be showing on your screen right now. I will do my best to answer some questions at the end of the presentation. This webinar, including the Q&A session, will be archived on our website at www.genscript.com/webinars.html. Also, you will receive a brief survey immediately after this webinar is over. I ask that you please consider filling it out, as your feedback is an invaluable resource for us to improve our services for you. So what is CRISPR? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, and the part of the CRISPR system is Cas9. Cas is an acronym for CRISPR-associated system, which is an RNA-guided double-stranded DNA binding protein with nuclease activity. Wild-type Cas9 is capable of generating a double-stranded cut that results in two blunt ends on the target DNA. As I mentioned in the beginning, CRISPR was first discovered as part of an adaptive immune system in bacteria. The type 2 CRISPR system from Streptococcus pyogenes is the one that gets transformed into genome editing applications in various organisms. The type 2 CRISPR locus contains the coding region for the trans-activating RNA, the Cas complex proteins, and a region that contains spacers segregated by direct repeats. When viral DNA enters the bacteria, the Cas complex recognizes the viral DNA and cleaves it into small fragments, which are then inserted into the CRISPR locus as spacers between repeat sequences. Spacers in this sense are stored as molecular signature of viral DNA. Once the viral DNA has been inserted into the CRISPR locus as spacers, it now can be transcribed together with the repeats as the pre-CRISPR RNA. Then trans-activating CRISPR RNA hybridizes to the repeat regions of the pre-CRISPR RNA, forming an RNA duplex that is then processed by endogenous RNA3 to generate the mature CRISPR RNA, trans-activating CRISPR RNA and the Cas9 complex. If the bacteria are exposed to the same virus again, the mature CRISPR-Cas9 complex will recognize the viral DNA based on RNA-DNA sequence complementarity and cleave the viral DNA by a double-stranded cut by Cas9 nuclease. The observation that CRISPR-Cas9 system is able to make double-stranded break on target DNA sequence guided by simple RNA-DNA sequence complementarity inspired scientists to engineer this system to perform genome editing in eukaryotes. In 2013, two science papers published simultaneously from George Church and Feng Zhang's lab demonstrate the ability of CRISPR-Cas9 system to perform genome editing in eukaryotic cells. The essential components of the engineered CRISPR-Cas9 system include the Cas9 nuclease and the guide RNA. The guide RNA consists of the guide RNA scaffold, which is of similar function as the transactivating CRISPR RNA, and a 20-nucleotide sequence, which mimics the function of the spacer. CRISPR-Cas9 complex recognizes the target site based on sequence complementarity with the 20-nucleotide target sequence in the guide RNA. The Cas9 nuclease will make a double-stranded cut only if a protospacer adjacent motif or PAM sequence NGG is presented at 3' of the target site. The double-stranded cut occurs 3 base pair upstream of the PAM site on the target gene. For adapting a CRISPR-Cas9 system in eukaryotic cells, we can use either an all-in-one plasmid or two plasmid platform. Here shows the two plasmid strategy for mammalian cells. In this platform, we have one plasmid encoding the human codon-optimized Cas9 with the SV40 nuclear localization signal driven by the CMV promoter. We have another plasmid encoding the guide RNA, which contains the target sites and the guide RNA scaffold, driven by the U6 promoter. In the all-in-one plasmid platform, both the Cas9 and the guide RNA are constructed in the same vector, driven by different promoters. Once Cas9 nuclease has generated a targeted double-stranded break on the gene of interest, we can then rely on host DNA repair machinery to achieve targeted genome editing. There are two pathways for DNA repair. On the left is the pathway for non-homologous end joining, or NHEJ, which is an error-prone pathway. When DNA is repaired under this pathway, the two ends simply rejoin together. Random insertions or deletions, also known as indels, are introduced at the site of repair, eventually leading to frame shift mutations and gene knockout. On the right side is the pathway for homology-directed repair or homologous recombination (HR). When DNA is repaired under this pathway, we can utilize an exogenous donor template to achieve knock-in. The donor template can be either a single-strand DNA or double-strand DNA that has homologous arms to the regions around the double-stranded break. Besides, the donor template should also contain desired elements we want to introduce into the target gene, like a single point mutation, a bunch of point mutations, or even a piece of insert. Cells utilizes the information on the donor template to repair the double-stranded break, incorporating changes on the exogenous donor template into the endogenous copy of the target gene, and leading to precise alteration, correction, and gene knock-in. CRISPR-Cas9 has many potential applications in research and therapeutics because of its simplicity. The hottest application is, of course, genome editing, where CRISPR-Cas9 has the advantage of being a very simple to implement system compared to other genome editing methods, such as zinc finger nuclease or TALENs. For genome editing, we can use the wild-type Cas9 to create double-stranded cuts, deletions between two cuts, or we can use the Cas9 nickase, which is a mutant version of Cas9 to create single nick or double nicks. Targeted genome editing can then be achieved by exploiting DNA repair pathways as described in last slide. There are also other interesting applications for CRISPR-Cas9 by utilizing the nuclease null mutant of Cas9, which allows Cas9 to become a vehicle to bring proteins or nucleic acid to target DNA regions. These fusion modules can be used to regulate, reorganize, and visualize gene, making CRISPR-Cas9 an even more versatile technology. Though there is a lot of potential for CRISPR-Cas9 being a powerful genome editing tool, there are limitations. Off-target is a major concern. The canonical guide RNA target sequence is 20 nucleotide long with a NGG PAM sequence adjacent at its 3'. Cas9 can still have nuclease activity even with 1 to 3 mismatches between the guide RNA and the target sites. This can potentially lead to off-target nuclease activity, and since Cas9 can induce double-stranded breaks, any off-target nuclease activity can cause mutations in those genes and may even cause oncogenesis. In the next session, I will walk you through the steps of guide RNA design and recommend actions at each step to help you design high-specificity guide RNAs based on what we've learned about the CRISPR-Cas9 system. Our goal is to find those accurate guide RNA target sites with lowest off-target potential and the optimum target location. To give you an overall picture, here I'm showing an overview of all steps involved in guide RNA design, starting from target gene analysis all the way down to guide RNA delivery. So next, let's walk through each step in great details. Step 1. Target gene analysis. The major purpose of this step is to make sure you get the accurate target gene sequence of your interest. There are multiple ways to get the target gene sequence. You can use the target gene sequence information in NCBI database, or if you want to check the possibility of gene variants, or if the sequence information is unknown for the species you work with. Genome sequencing of the target gene is always the best way. And we always recommend our clients to sequence the target gene in the cells or strands you work with before designing guide RNA. This is particularly important for knock-in experiments because you will need accurate sequence information to design homologous arms for your donor template. Also, gene structure needs to be analyzed to know locations of exons or coding DNA sequence CDS and the introns. Such information is important for selecting optimum target location, which I will cover in step 4. Step 2. Find guide RNA canonical sequences based on the accurate target gene sequence identified in step 1. We know that the canonical guide RNA sequence is 20 nucleotide long with an NGG PAM sequence adjacent to its 3'. In mammalian system, we usually use the U6 promoter to drive the expression of guide RNA, and the G is the first nucleotide for U6 promoter binding and recognition. So the consensus guide RNA sequence becomes GN20GG. One additional note on the PAM sequence. So the canonical PAM sequence is NGG in CRISPR-Cas9 system. NAG is also considered to be a PAM, but we always use NGG for better targeting efficiency. NAG PAM sequence will be checked when evaluating off-target effects. After finding all canonical guide RNA sequences in the target gene, the next step is to eliminate those guide RNAs with high off-target potential and return the ones with low off-target effects. For each guide RNA, you will need to blast the GN20 GG sequence against the whole genome to find the similar sequences. Of course, the higher similarity, the higher off-target risk. But how can we carefully gauge the off-target risk? We know that 20 nucleotide plus NGG PAM sequence adjacent to 3' compose the guide RNA target site. In addition to that, according to a seed model, the 20 nucleotide guide RNA target site can be divided into two parts. The 12 nucleotides adjacent to the PAM sequence, referred as the seed sequences, which are most critical for binding and cleavage by the Cas9 nuclease. So any sequences in the genome that shares high similarity with the seed sequences will have higher off-target potential. Based on all those findings, we summarize 3 criteria for gauging the off-target risk. First, existence for PAM sequences, both NGG and NAG at 3'. Second, sequence similarity. The greater the number of matches, the higher risk of off-target binding. Third, sequence similarity at 3' region increases off-target risk. So after this off-target analysis, we will probably have several guide RNAs left with low off-target risk. The next step, step 4, is then to choose the guide RNAs with the best locations to meet your research purpose. If your goal is to knock out a gene, we suggest targeting early exons, because knockout is achieved by exploiting the NHEJ repair pathway. If indels are introduced in early exons, you will have a higher chance to obtain reading frame shift mutants. If you want to knock out the function of a specific protein domain, guide RNAs targeting the specific domain will be selected. For knock-in purpose, usually the target region is designated without many choices. Therefore, we simply find guide RNA canonical sequences within the targeting region. Most likely, we don't have many guide RNAs to compare. In this case, we just choose the relative good ones based on off-target analysis. Both exon and intron can be targeted. Selection of exons for guide RNA targeting is mostly due to the design of donor template. If guide RNA targets an exon, when we design the donor template, the guide RNA targeting sequence can be mutated silently without changing the encoded protein. However, if the intron is targeted, mutations may be introduced into a targeted sequence to avoid miscleavage of donor template by guide RNA. So usually we mutate the PAM sequence NGG to NCG on donor template. After this step, you will have a few good RNA candidates left. We recommend choosing at least 2 guide RNAs for each target gene, though the efficiency of CRISPR system is very high. Now you have selected the best guide RNAs that can potentially meet your research goals. The next step is to deliver the guide RNAs into cells. Guide RNAs can be delivered to cells as a DNA plasmid, as RNA, or transgenically. Delivery of guide RNA expression plasmid or RNA is very efficient and can be generated in a very short time frame. The transgenic source of guide RNA is of very high efficiency, but it's very expensive and time-consuming. The most common way we use for mammalian system is through guide RNA expression plasmid, in which it contains a U6 promoter that drives the transcription of guide RNA, the guide RNA targeting region, the guide RNA scaffold, as well as a termination signal for guide RNA transcription termination. The guide RNA expression plasmid can be an independent one for guide RNA expression only, or can be an all-in-one plasmid co-expressing the Cas9 nuclease. So far, we have gone through all the steps involved in guide RNA design. You understand that off-target risk is a key issue to address when designing guide RNAs for CRISPR-Cas9 system. Several approaches have been discovered to enhance the targeting efficiency by modifying either the guide RNA or the Cas9 nuclease. One study determined the effect of truncating the guide RNA. By targeting EGFP, researchers were quickly able to determine that guide RNA sequences between 17 and 20 base pairs in length achieved similar levels of on-target gene editing. At 16 base pair, there was very little indel activity. Truncated guide RNAs that were 17 or 18 base pairs in length were used to target 3 different human genes, VEGFA, EMX1, and CLTA. The 17 and 18 base pair guide RNA sequences improved specificity up to 10,000-fold. So the key takeaway from this paper is that it is beneficial to keep the targeting sequence of the guide RNA at 17 or 18 bases to reduce potential off-target effects. Another approach is to modify the Cas9 nuclease. Two modified Cas9 nuclease systems have been developed to reduce off-target effects. One system is referred as the Cas9 nickase system. In this system, the D10A Cas9 mutants allows for single-strand nicking. Since Cas9 nickase only nicks a single strand, 2 Cas9 guide RNA complex would be needed to generate a double-stranded strand break. Therefore, 2 guide RNAs would need to be designed. And the guide RNA would require an offset, which is the distance between the 2 guide RNAs, to generate a 5' overhang. Using this system, researchers were able to observe a 1,500-fold increase in specificity compared to using a single guide RNA and wild-type Cas9. The other modified system is referred as the dimeric CRISPR RNA-guided fork-1 nuclease system. In this system, fork-1 nuclease, which is used in the TALEN genome editing system, was fused to a catalytically inactive Cas9 mutant, or dead Cas9. Fork-1 nuclease is introduced to increase specificity because it only has nuclease activity when it's in a dimer form. Similar as the Cas9 nickase system, 2 guide RNAs would be needed to allow 2 Cas9 fork-1 complex to bind the target DNA and dimerize. Using this system, researchers found out that it has up to 10,000-fold less mutagenic activity compared to a single guide RNA and wild-type Cas9. To design guide RNAs for the Cas9 nickase or the fork-1 dead Cas9, you can adopt the similar principles we described for wild-type Cas9, with attention to 2 special things. First, you need to design 2 guide RNAs to target each strand of the target gene respectively. And second, when designing paired guide RNAs, you need to consider the offset, which is the distance between the 2 guide RNAs. Based on literature and our experience, for the Cas9 nickase system, we suggest -4 to 20 base pair offset. A wider range of -8 to 100 base pair is also mentioned in literature. For fork-1 and dead Cas9, we suggest 14 to 17 base pair offset. So to make the guide RNA design work easier, scientists at GenScript developed a leading guide RNA design tool and made it freely online available to the entire research community. This design tool can be accessed at genscript.com/guide-RNA-design-tool.html. Several features about this tool. First, this tool works for designing guide RNAs for the wild-type Cas9 nuclease system, as well as the Cas9 nickase system. Second, it accommodates multiple target input options, which means you can design guide RNAs by providing gene name, gene sample, gene ID, or gene sequences. Third, you can use this tool to design guide RNAs for most popular mammalian systems, including human, mouse, and CHO cells. Another unique feature about this tool is that we provide off-target risk scores based on sequence similarity throughout the genome. Risk scores range from 0, the best, and to 58, the worst. We recommend selecting guide RNAs with risk score less than 49 to avoid possibility of off-target binding. This unique feature will enable you to easily select the guide RNAs with high targeting specificity. Besides the free guide RNA online design tool, we also offer two specialized CRISPR services at GenScript to meet researchers' needs. The guide RNA construct service gives our customers the opportunity to take advantage of our best-in-class gene synthesis and molecular biology, as well as our scientists' expertise in designing guide RNAs. This service covers both design and construction. It starts with the critical step of guide RNA design, where our scientists will carefully design the guide RNAs targeting your gene of interest. Using our best molecular biology platform, we will synthesize the guide RNA targeting sequence, clone it into the guide RNA vector, and deliver you the ready-to-use guide RNA expression plasmid within 10 business days for just $1.99 per construct, which is the lowest pricing available for full guide RNA design and construct service. For this service, we offer a standard guide RNA cloning vector for free. This vector is suitable for use in any mammalian system. We can also subclone guide RNAs into your own vector at no additional cost. If you choose to use our free guide RNA vector, we will also supply the human codon-optimized Cas9 expression plasmid for free. We keep optimizing our CRISPR platform. A set of in-house developed all-in-one vectors are available now, so please check back often as we will be constantly adding additional offerings to the guide RNA construct service. The GenCRISPR cell line service is a full CRISPR-based genome editing service to produce genetically modified cell line using any mammalian cell line and targeting any gene. This is a comprehensive service that starts from guide RNA design to transfection and single clone generation of a wide range of cells, including difficult to transfect and tumor cell lines. We will deliver to you a target sequence validated single clone with the genome modification specified, along with a detailed report on generation of the clone. So all the work will be done by a dedicated group of CRISPR experts at GenScript. We can also perform validation assays for you. Our in vitro pharmacology services offer many validated cell function assays, including in-cell Westerns, GPCR and ion channel assays, growth and apoptosis assays. And we can also provide off-target analysis using Sanger or next-generation sequencing for deep sequencing analysis. We provide both CRISPR knockout and knock-in cell lines with pricing at $8,000 and $9,000 respectively. We are running a promotion for the cell line service in November. You are eligible to receive a 30% discount of the listing price in this month. So please catch this great opportunity if you need knock-in or knock-out cell lines for your research. To wrap up today's webinar, I'd like to summarize the key points. The simplicity of CRISPR-Cas9 system makes it an efficient and easy to implement system for genome editing, giving more researchers the opportunity to use genome editing technologies in their labs. However, a major concern in using CRISPR-Cas9 is the potential for off-target effects, since the system can tolerate 1 to 3 mismatches. Careful guide RNA design, starting from target gene analysis to find canonical guide RNA sequences, off-target analysis, location analysis, and guide RNA delivery allows us to obtain guide RNAs with lowest off-target risk. Modified Cas9 nucleases, including Cas9 nickase and FOC1 dead Cas9, can improve specificity. Paired guide RNAs are needed for modified Cas9 systems. GenScript offers a free guide RNA online design tool featuring on off-target risk ranking. And finally, we hope you will consider our GenCRISPR services for your research needs. We offer a complete genome editing solution, including guide RNA design and construct service, as well as custom cell line development. So I would like to thank everyone for taking the time to join this webinar. I hope this has been useful to you and that you can apply some of what I talked about today to your own CRISPR genome editing projects. Now I will answer questions you have. So the first question is, can we get the slides for this presentation? Yes. The recorded webinar along with the slides will be archived on our website at genscript.com/webinars.html. You can download the slides approximately 1 week after the webinar, and also you can directly email me for the slides. The second question we have is, do you have the nickase Cas9 form? Yes, so we have the nickase Cas9 expression plasmid ready at GenScript, and you can request for that. The next question we have here is, when design donor template, how long should the homologous arm be? So this depends whether your donor is a double-stranded or single-stranded donor. So for double-stranded donor, we recommend 500 to 1000 base pair. And some literatures show homologous arms around 1000 base pair is required. And single-stranded donor can be shorter than that. And the next question is, do you offer services for organisms other than mammalian cells? Yes. The guide RNA construct service can be offered to different organisms, including mammalian, bacteria, plant, Drosophila, etc. And our standard vectors are for mammalian cells only, so you will need to supply the guide RNA and the Cas9 expression vectors for other systems. And the cell line service is for mammalian system only. And the next question is, which system is better, 2 plasmid or all-in-one plasmid? Based on our experience, both systems are of similar targeting efficiency. We have both platforms ready at GenScript, and the 2 plasmid platform For this two plasmid platform, we recommend a 1:1 transfection ratio. And the next question is, can I use CRISPR on primary cells? Yes, primary cells can be done if the purpose is to obtain a knockout cell pool. Since most primary cells have limited lifespan, so isogenic clones are difficult to obtain. And the next question is, are you developing an online tool generating 17 to 18 base pair guide RNA sequences? The answer is yes. That's in our R&D developing pipeline. So we are working on that, and please closely watch our updates. We probably will send out an email if this tool is available. And the next question is, do you have any experience working on plants? Yes, we have helped many plant biologists design guide RNAs. We also synthesized, codon optimized the Cas9 genes for our clients. So you can find some related publication in our citation database. But currently, we do not offer standard guide RNA expression vector for plants. You may need to supply the vector if you choose to use the guide RNA construct service. And the next question is, can I generate conditional knockout cell line using CRISPR? Conditional knockout cell line can be generated by combining with other elements, for example, the Cre-LoxP system. However, integration of LoxP sites into host genome is a process of knock-in. And we have another question coming up. So what should I provide if I want to use the guide RNA construct service from you? That's very simple. You just need to provide the accurate target gene sequence and specify how many guide RNAs you need. We will design the guide RNAs according to your specification. You can make an online submission of your request, which is very convenient, or directly contact the tech support at GenScript. We provide standard expression vector for free, and if you want to clone the guide RNA into expression vector other than the standard vector, you can just send us a small aliquot of that vector. And the next question is, will copy number of target gene affect genome editing? Yes, increasing number of gene copy reduces the chance to obtain homozygous modification of the target gene. So which means that the higher the copy number of your target gene, the lower chance you will obtain homozygous modifications. And the next question we have is, are the Cas9 optimized for zebrafish? I believe we have done this for our clients, and also you can send in codon optimization requests for us, and we can optimize the Cas9 for zebrafish for you. Okay, so I will answer one last question for today. Is it possible to design guide RNA on intron region of a gene or a non-coding region? Yeah, you can find. Yes, you can design guide RNA to target intron regions if you can find the canonical guide RNA sequence, which means a PAM sequence NGG exists, and the guide RNA targeting sequence can be identified. But for the knock-in experiments, you will need to introduce mutations to the guide RNA target sequence to avoid the mistargeting of donor template by guide RNAs. Okay, so that's about what we have for today. For those unanswered questions, I will post the answers to our website together with today's webinar presentation and Q&A. And you can also email me directly at heidi.huang@genscript.com with specific questions about this presentation. So also please take a moment to complete a quick poll right after this webinar and tell us how you liked this presentation. Your feedback is very helpful for us to improve our services. So again, thank you so much for joining us today, and good luck with your future research.